Ovulation is triggered by a surge in luteinizing hormone, leading to enzymatic degradation and rupture of the follicular wall. The secondary oocyte, encased in a protective corona radiata, is released into the peritoneal cavity. The time-lapse animation shows follicular maturation inside the ovary, highlighting the vascular network. Include the roles of estradiol buildup and LH receptor expression in theca interna and granulosa cells. The infundibulum's fimbria, lined with beating cilia, generate directional currents that capture and transport the oocyte into the fallopian tube lumen. The animation illustrates microvilli on fimbria tips and peristaltic muscular contractions of the infundibulum wall. Explain how local prostaglandins influence ciliary beat frequency and oviductal secretions. Capacitation alters the sperm membrane, enhancing motility and responsiveness to oocyte signaling. Biochemical changes prepare the sperm for zona pellucida binding and acrosomal exocytosis. Describe hormonal triggers and survival rates during sperm ascent. Sperm bind specific receptors on the zona pellucida, initiating the acrosome reaction. Hydrolytic enzymes enable penetration through glycoprotein layers toward the oocyte membrane. Discuss molecular recognition between sperm membrane receptors and ZP glycoproteins. Fusion triggers calcium oscillations in the oocyte, causing cortical granule exocytosis. This modifies the zona pellucida to prevent polyspermy, preserving genomic integrity. Emphasize signal transduction cascades initiated by sperm-oocyte fusion, such as PLC-zeta. The oocyte completes meiosis too, expelling the second polar body. Meanwhile, the sperm nucleus decondenses and both pronuclei migrate, preparing for syngamy. Mention the reactivation of metaphase arrest via MPF deactivation. Pronuclei merge as spindle fibers align chromosomes along the metaphase plate. The maternal and paternal genomes combine, forming the diploid zygote nucleus. Mention epigenetic reprogramming events, DNA methylation reset, and genomic imprinting preservation. Mitotic cleavage transforms the zygote into a multicellular structure. Cells retain the zona pellucida for protection as they advance toward the uterus. Detail cleavage timing, maternal mRNA degradation, and activation of the embryonic genome. Conception is the culmination of synchronized hormonal cues, cellular transformations, and molecular signaling, resulting in the creation of a single totipotent cell, the zygote.